Hello, everyone. I'd like to first thank the Botany 2020 organizers for putting this together and keeping the spirit of science alive during these trying times. My name is Brian Riatini, and today I'm going to be talking about some work that I've been doing down in the Galapagos Islands, investigating the contribution of reproductive interference to the local extinction of an endemic Galapagos tree species, Psidium galapagos. The introduction of plant species to regions where they don't occur natively has dramatically altered plant richness patterns around the world, and islands are certainly no exception. For example, in the Galapagos Islands, there are actually now more introduced plant species in the islands than there are natives. And as a consequence, many formerly isolated native and introduced species have come into secondary contact. When native and introduced species come into secondary contact, they can interact in a number of ways that can jeopardize their long-term coexistence, and resultantly pose issues for the conservation of native species. For example, if competition occurs between them, it can lead to the local extinction of native populations via competitive exclusion. Alternatively, if frequent hybridization occurs between them, it can lead to the eventual replacement of native populations by hybrids. If instead heterospecific mating results in reduced parental fitness, such as if hybrids are inviable, then wasted reproductive effort can lead to the local extinction of native populations if reinforcement doesn't occur first. And today I'll be focusing on this last option, which is commonly referred to as reproductive interference. Of the many invasive plants in the Galapagos Islands, Psidium guayava, or common guava, is among the most problematic and longest standing. Since its original introduction to San Cristobal Island in the late 1800s, it has spread like wildfire throughout the highlands of the human-inhabited islands, where it has come into secondary contact with an endemic species of guava, Psidium galapagos, which is known locally as guayabillo. And as we can see here in these maps of the predicted ranges of the two species, where guava is displayed in pink and guayabillo in blue, there is a large region of predicted sympatry between the two on the co-inhabited islands, which is here shown in purple. However, if we zoom in on San Cristobal Island, where the guava invasion began over 100 years ago, we can see that the extant populations of guayabillo, shown here as white dots, occur only near or beyond the edge of guava's predicted range. And this is strikingly consistent with guava's actual distribution on this island, as we can see here in this land cover map constructed using satellite and drone imaging, where guava is displayed in yellow. So why is guayabillo not found throughout much of predicted sympatry, whereas guava dominates predicted sympatry? One potential explanation is that guava has already displaced guayabillo from these regions, and that sympatric guayabillo populations at lower elevations, where guava has become established more recently, may be under threat of local extinction. And that brings us to the question at hand. Are negative interactions such as reproductive interference occurring between these species? And if so, might it be contributing to the apparent decline of guayabillo populations? In order to investigate the potential for reproductive interference between these species, in 2018 and 2019, I went down into a few contact zones and evaluated the strength of pre- and post-zygotic barriers between these species. And what I found is that pre-zygotic barriers appear to be weak. Specifically, both species flowered at the same time in sympatry. They were also pollinated by the same species, particularly by the Galapagos carpenter bee, which is quantitatively the most important pollinator on the islands and the most generalized, and was also the most frequent pollinator for both of these species in sympatry. And then finally, when pollen from invasive guava is transferred to flowers of guayabillo, as I did by hand, it can germinate and develop pollen tubes which traverse the style and enter the ovary, where interspecific fertilization may then take place. However, it appears that such interspecific fertilization is quite literally a dead end. I performed controlled crosses between these species and found that interspecific crosses resulted in a reduction of fruit set relative to intraspecific crosses and self-fertilization. Moreover, the fruit that was produced by interspecific crosses when guayabillo was the maternal parent contained only inviable seeds. And although we're not sure yet what the genetic mechanism of post-zygotic isolation is, we suspect that it may involve differences in ploidy. We have some preliminary genetic and genomic evidence that suggests that guayabillo may be polyploid, whereas guava is diploid. And commonly in plants, interploidal crosses can produce seeds with reduced endosperm size and resultant inviability when the maternal parent has the higher ploidy level. And this could potentially explain the shriveled and inviable seeds that we recovered from the interspecific crosses when guayabillo was the maternal parent. In order to verify that the ongoing production of viable hybrids is not occurring under natural conditions, we surveyed secondary contact zones across 3 of the human-inhabited islands for genetic evidence of hybridization. Using 9 microsatellite loci, we analyzed a total of 269 guava individuals and 135 guayabillo individuals, including some putative hybrids. However, all ended up having a posterior probability of less than 1% of being a hybrid, and it was clear that no early generation hybrids were being produced in these contact zones, at least at detectable frequencies. And this again suggests that post-zygotic barriers between these species are strong, even under natural conditions. And this combination of weak pre-zygotic barriers and strong post-zygotic barriers may foster conditions for reproductive interference for both species, but why might we expect it to have a greater impact on guayabillo than guava? Why didn't guayabillo simply prevent the establishment of guava in the first place, for example? Well, guava, as it turns out, can reproduce asexually, and it typically does so by forming dense clones. To our knowledge, guayabillo can't do that. So, although reproductive interference may negatively impact sexual reproduction for both of these species, guava doesn't rely on sexual reproduction to spread, whereas guayabillo does. And this could cause some serious problems for guayabillo populations, given that a portion of native embryos may be wasted on producing inviable hybrid seeds. And if reproductive interference is strong enough, it may lead to the demographic decline and eventual local extinction of some of these guayabillo populations. And it appears that that may be the case. Both fruit set and seed set for guayabillo were significantly lower in sympatry relative to allopatry, which is consistent with our model of reproductive interference. And given this reduction of viable seeds that are being produced in sympatry, we then predicted that there would consequently be fewer seedlings and juveniles in those sites, which could be a sign of demographic decline. And so I used tree height as a proxy, a rough proxy for age, with the expectation being that sympatric populations would have fewer seedlings and juveniles, which would be reflected in having fewer smaller individuals than allopatric populations. And that's exactly what I found. There were actually only 2 individuals less than 2 meters of height across both sympatric sites, whereas individuals of these smaller size classes were abundant in allopatry. Notice also the stage of demographic decline appears to be more pronounced for sympatric LG than sympatric CM, which is consistent with LG having lower seed set. Moreover, it is consistent with studies that have shown that the strength of reproductive interference depends on the abundance of the interfering species. If we look at land cover maps on San Cristobal again, we can see that sympatric LG is in a region of high guava density here. The light pink is displaying guava-dominated forests, whereas the purple is showing mixed forests. And we can see that CM is in one of these areas of mixed forests where guava is not yet the most dominant species. Thus, the lower reproductive success and more apparent demographic decline in LG versus CM appears to be consistent with the fact that LG is in a region of high guava density and therefore in a region where reproductive interference should be stronger. And so this doesn't bode well for these sympatric guayabillo populations. If there are no seedlings to replace the remaining adult guayabillo individuals when the adults die, local extinction of these populations might be inevitable. And given that guayabillo is already limited to the edge of guava's range on San Cristobal, it appears that the range decay of guayabillo on this island may be approaching equilibrium. But San Cristobal was the first island to be colonized by guava, which begs the question, how might this pattern differ on islands where guava was introduced more recently? Our prediction given these data would be that guayabillo would occur more continuously throughout predicted sympatry on islands where the guava invasion is more recent. And that does appear to be the case. Here I've plotted the relative position of each Guayabillo population, represented as these blue dots, between the centroid of sympatry, which is at 0, and the edge of predicted sympatry, which is at 1. And as we can see, it appears that Guayabillo populations occur more continuously throughout predicted sympatry on Santa Cruz and Isabella, which are the most recent islands to be colonized by guava. Thus, there appears to be somewhat of a relationship between the extent of Guayabillo's decline and the age of the guava invasion on that island. Although without human intervention, local extinction of Guayabillo populations within the highlands of these 4 inhabited islands may be inevitable, archipelago-wide extinction is probably unlikely. And this is because Guayabillo populations remain outside of the predicted range of guava on the co-inhabited islands, such as here on Isabella, and there are actually also a few isolated islands populated by Guayabillo that are also outside of the predicted range of guava, such as Fernandina, Santiago, and Pinta. These islands may offer sanctuaries from this invasive species even after the guava invasion has reached the limit of its range expansion, at least barring any further adaptation to the local environments. In conclusion, the combination of weak prezygotic barriers and strong postzygotic barriers appears to be fostering conditions for reproductive interference in the system. Within sympatric Guayabillo populations, Guayabillo has reduced fruit set and seed set and shows signs of demographic decline. And the stage of Guayabillo's decline on each island appears to depend at least somewhat on the age of the guava invasion on that island. And this is rather interesting because not only does it appear that we've caught this reproductive interference leading to local extinction as it is in process, but that there are multiple naturally occurring experimental replicates on the different islands, each at a different stage of decline depending on the age of the invasion on that island. And finally, although local extinction within the highlands of these human inhabited islands appears to be inevitable without human intervention, archipelago-wide extinction of this protected Galapagos endemic is unlikely. So there is a silver lining for Guayabillo. And with that, I'd like to thank a whole horde of people, both from UNC and USFQ, the Galapagos Science Center down in Galapagos, in addition to a few of my funding sources. And with that, I'd be happy to take any questions that you may have, either live here or via email. Thank you very much.